Thank you. Okay, before coming to my lab, Michael earned his master's and PhD from the Hebrew University of Jerusalem, where he worked with a biologist, Ronen Kedman, and physicist Nadav Schnurb. And Michael's already published a great deal for his career stage, even with more publications from his dissertation on the way. And it includes publications in widely read journals like PNAS, Ecology Letters, and American Naturalist. In my lab, Michael's carrying out analyses of the forest census data on Barra Colorado Island in Panama to look at spatial patterns indicative of competitive coexistence mechanisms among tree species, and to also fit demographic models of them to estimate their influence on tree diversity. And he's been getting interesting results on that, but I'll probably present that at a future seminar. And I'll say it's really been a pleasure working with Michael so far. I'm impressed with the breadth of topics in ecology he has a deep understanding of, and the many skills he has command of, and also the care that he takes to be rigorous in his work. So today he's going to tell us about research from his PhD dissertation, where he tested community assembly theory predictions of dynamical patterns, where most tests of community assembly theory have been focused on static patterns. So with that, I'll turn it over to Michael.
Okay, Annette, thank you very much for the introduction, and thank you for joining in. And thank you for this opportunity to tell you something about the things that I've done during my PhD with Ronen and Nadav. But before that, let me start by saying that when I came here to EEB in the University of Michigan from my original university with our 8 EEB faculty, I was so excited to come to a really big international department of EEB with so much interesting things going on, so many interesting people doing interesting research. And I really wanted to get to know people and to get to know what people are researching. But let us say that circumstances were not in my favor. So at least I hope that with this talk, you will at least know a little bit about what I've done. And please view this as an invitation to talk about shared interests or whatever. Please contact me. I'll be very, very glad. Okay, so I'm going to tell you about some perhaps small steps I have taken towards bridging the gap between statics and dynamics in community ecology. I'll begin by explaining you what I mean by the gap between statics and dynamics. And I hope that by the end of the talk, I'll convince you that it is an interesting and worthwhile direction to pursue. But let's begin with a basic and simple question. How do we understand the roles that different processes play in shaping ecological communities? How do competition, dispersal, predation, whatever, shape ecological communities? One option is to do experiments, but this unfortunately is not always practical. What people often do is they study observed patterns in natural ecological communities, and many of these patterns reoccur in many communities. For example, the dependence of the number of species on area and on isolation, the number of common and the number of rare species. And the link between the patterns and the processes is often done through a dynamic model, basically asking are the changes in the community influenced by the community itself and the environment. And the dynamic model makes assumptions on, it incorporates the processes through its assumptions. It assumes that competition does that and dispersal does this. And at the end of the day, if the model successfully explains the patterns, then this can serve as some evidence to the success of the model and to the importance of these processes. So in the beginning of my career, I studied this kind of patterns, but after a while looking at them, I thought there's some recurring motif to these patterns. They're all static. They all describe a fixed snapshot, a still image of ecological communities, which made me ask, okay, can these models that represent our understanding of how ecological communities act, can they also explain dynamic patterns patterns of change, temporal patterns, such as how do the abundances of species change with time? How does the number of species change with time? And when I started reading about it, I found out that, first of all, not so much is known about temporal patterns at a community level. I mean, there have been works, but not as many. And even less is known, I think, about the ability of different dynamic models to explain these patterns. And I think it's unfortunate because it does make sense that the dynamics that we observe in ecological communities can teach us quite a lot about the ecological processes as they act through time to shape ecological communities. So with this, the goal of my PhD work, and today I'll tell you about my PhD work, was to promote the understanding of the processes shaping ecological communities by relating dynamic patterns with dynamic models. And in particular, I asked for the two most minimalistic models of community dynamics, and I'll in a minute explain what I mean by this term, neutral theory and dynamic equilibrium. First, can the model explain observed patterns of community dynamics? And second, if not, then what processes do we need to add so that we can explain community dynamics? Before moving on, what do I mean by minimalistic models of community dynamics, such as neutral theory? The minimalistic models, I think, have a dual role. On the one hand, they are dynamic, full-blown dynamic models. So they can make many predictions about how various properties of the system will change with time, and how will they look in the future, how will they look in steady state, a lot of possible predictions. On the other hand, they are minimalistic, and that's in the sense that they incorporate only the bare bone, most simple processes that you can have. And as a result, if there are deviations from the model, it means that something is missing in the model. Not something is excessive, but something is missing. And this deviations can kind of hint what is that missing part in the model. So in other words, they can be used as dynamic null models for ecological communities. So after this brief introduction, my talk will have two parts. In the beginning, I'll talk about the neutral theory of biodiversity. I will present the theory, how I tested its dynamics. Then following the test, I developed an extension of neutral theory, which I will describe. And then I'll describe the test of this extension, make some conclusions, and then I'll move to the dynamic equilibrium theory, which is another name for island biogeography. I'll go into this later. I'll explain what it means, what the theory says, how it was tested, and then I'll present a new methodology that I developed to testing and quantifying deviations of communities from dynamic equilibrium. Then I'll present to you an analysis of thousands of communities showing pretty much the same patterns, and then I'll make some broad conclusions, perhaps. So stay tuned. Let's begin with neutral theory. The Unified Neutral Theory of Biodiversity and Biogeography, impressive name, NTB, pretty much originally was developed to explain patterns of species richness, commonness, and rarity. It works the following way. Suppose we have a local community of J individuals belonging to different species. Every time step, one individual dies and is immediately replaced by an offspring. The question is, who's the offspring? So its probability M. This offspring is of some parent that is somewhere in the meta community, the big, large species pool surrounding the local community. And with probability 1 minus M, there is a local competition. So we pick the parent at random. And the offspring has the same identity as the parent. And that's pretty much everything that happens in neutral theory. So this is kind of an algorithm, a computer game. Let's talk more ecology. What's going on here? So the processes in neutral theory are basically a random birth, death, and immigration events. There's some form of dispersal limitation. Not everybody not all the offsprings are produced locally. And there is competition, but it is symmetric. And that's a core assumption of neutral theory, neutrality or symmetry, meaning all the species are exactly the same. And this crucial simplifying assumptions make this theory extremely simple and minimalistic. basically no differences between species. I guess it's very natural to ask, what can such a simplistic theory possibly explain? And it turned out that neutral theory has an impressive ability to explain the distribution of commonness and rarity of species. These are in many communities. So here we see the species abundance distributions of a few coral reefs and tropical forests, and we'll be seeing a few of those, so pay attention. Always in these figures on the y-axis, we have the number of species whose abundance is in the local community, whose abundance is given on the x-axis on a log2 scale. So for example, in the tropical forest of Korup, there are 50 species with around 2 to the 5th power individuals. The black curves are the fifths of neutral theory. And in all cases, they are pretty remarkable. So overall, it's possible to say neutral theory has had impressive ability at explaining various static biodiversity patterns. But what about the dynamics? Does neutral theory explain the dynamics? Don't forget, the dynamics, the changes, is the thing that creates the statics on the long run. On the long run, these changes will shape the patterns that we see of commonness and rarity. So can neutral theory explain the dynamics? On long timescales, it has long been known that common species will have unrealistically long times till they go extinct. On average, a population of n individuals is expected to go extinct in 2n generations. So if we have 5 million trees from some species in the Amazon and the generation time is 100 years, we easily get to an extinction time of a billion years, which is a little bit too much. What about shorter timescales, ecological timescales that we see in surveys with our own eyes? Here, until recent years, not too much has been known. So the aim of this part of my work was to test the ability of neutral theory to explain observed dynamics. And when I say observed dynamics, it's important to clarify what do I mean? I mean this kind of data. You have the composition or the abundances of a bunch of species through time. And does this highly dimensional thing, can it be described by neutral theory? So first of all, what we need is, yes, accurate information of abundances through time, like this table here. So a good place to start would be the home court of neutral theory, the tropical forest plot in Barra, Colorado Island, Panama. In this plot, the populations and even the exact locations of individuals of 320 species of trees and understory plants are surveyed in a rectangle 500 by 1,000 meters every 5 years. And when I did the analysis, there were 5 surveys available. So can neutral theory explain the dynamics in BCI? Well, as I said, because this is a very complex thing, a matrix of species by years, we should kind of simplify it to manageable, understandable patterns that we can see by the eye. So what fundamental temporal patterns can we think about? Well, one option is that we know that the populations in the forest change all the time. Some grow by a factor 2, some decline by a factor 3. So it's possible to just plot a histogram of the distribution of population changes in 20 years on a log scale. And you can see that neutral theory in red, well, is not a very good description of BCI in blue. There is this extremely pronounced peak around 0 change. And this p-value means that there's basically no way of getting the forest under neutral theory. Okay, what other patterns can we look at? Another idea is that basically, as time goes, we can look at the level of species composition. As time goes by, it makes a lot of sense that the composition of the forest will become increasingly different from the initial composition. So we can do this by calculating, for example, the Bray-Curtis dissimilarity of every survey to the first survey. And indeed, neutral theory predicts that as time goes by, the community would become increasingly dissimilar. Only it's nowhere near what we see in BCI. The changes in BCI are much bigger. So these patterns and a few others make me conclude that neutral theory cannot explain the dynamics in short and long temporal timescales. And as I said, at the end of the day, the dynamics, the changes are what's going to, at the end of the day, make the static picture that we see. So if the simple null model fails, it means something is missing. And I hypothesized that if we add environmental fluctuations to neutral theory, this can fix the dynamics by introducing faster changes. First of all, it makes a lot of sense because we all know that the environment in which species live changes all the time. So it's a sensible hypothesis. And the questions are, first of all, will this work? Will we get a reasonable description of the dynamics? The second question is, okay, will we still be able to explain the species abundance distribution, which is the great achievement of neutral theory? So to test this hypothesis, we derived a model that is an extension of neutral theory. In this extended model, individuals still die at random, but now every species has some fitness. And the recruitment of new individuals depends both on fitness and on abundance. You can think about this fitness as basically proportional to the number of seeds a tree of some species produces. And it represents it's a very simple way of representing the response of the environment. If my fitness now here is high, then I like this environment. Importantly, every tau time steps, the environment changes. I don't know, it becomes drier. All the fitnesses then are redrawn from the same distribution with a standard deviation of A. And this parameter A is the magnitude of environmental fluctuations. If it is 0, then we go back to neutral theory. All the species have exactly the same fitness all the time. A is larger than 0, then the fitnesses of different, so at this time point, there is a competitive hierarchy, but then after the environment changes, there's a new competitive hierarchy, and the environment all the time changes, the fitness change. But on the long run, all the species still are the same on average. That's why we call it the time average neutral theory of biodiversity, or TNTB. Now that we have a model, let's test it against its null model, neutral theory. And let's see if TNTB can outperform neutral theory in explaining the short-term dynamics. Can it still explain species abundance distributions? And finally, perhaps, can the extended theory also predict more realistic times to the extinction of common species? So let's go back to BCI. And here again is the distribution of population fluctuations on a log scale on 20 years. BCI is in blue, and you can see that TNTB in green, the extended theory, is a much better fit to BCI than neutral theory in red. And in principle, the deviation between the forest and the theory is not even significant, which means there is actually a probability of getting the changes in BCI under the theory. Looking again at the changes in composition in time, both neutral theory and the extended TNTB predict that the similarity of the composition with respect to the first survey will increase with time, but only TNTB predicts anything close to what's going on in BCI. So these patterns and others make me conclude that TNTB explains the observed dynamics much better than its now neutral theory, and not bad in absolute terms. That's nice, but what about the species abundance distribution? Can we still maintain the success of neutral theory? Well, again, we see on the y-axis the number of species whose abundance is given on the x-axis. And despite some differences in explanatory power, they're both good and pretty similar. It's important to add that all the patterns you have seen have been fitted with the same parameters, representing our understanding of how often and how strongly does the environment change, how much dispersal there is. Fitting every pattern with the different parameters is cheating. Okay, so I started with saying that neutral theory predicts very long times until common species go extinct. Can the TNTB solve this issue, at least to some degree? Unfortunately, I don't have information about long timescales, so this analysis is purely theoretical. But when I simulated under various regimes, populations with different initial abundances, and saw how long it will take them to go extinct, you can see that for that neutral theory in red with a fixed environment is qualitatively different than all the situations in which the environment is fluctuating, which and common species go extinct considerably faster, which suggests that indeed neutral theory has the extended TMTB has the potential to address this problem as well. So to conclude the results of this part, I have shown that in neutral theory, in BCI, neutral theory can explain the species abundance distribution, and pretty much that's it. While the extended TMTB, the time average neutral theory, can explain both the species abundance distribution and fundamental patterns of dynamics of populations and the entire community composition. Also, I've shown you with the simulations that neutral theory predicts unrealistic time to extinction, while TMTB predicts probably more realistic time to extinction, although it's hard to quantify exactly. And finally, I want to add a small topic. I haven't talked about it before, so you're more than welcome to ask me later. There's another big problem with neutral theory. Neutral theory is very, very sensitive. The predictions are very sensitive to the assumption that all the species have exactly the same fitness on average on the long run. If even one species is a little bit better than others, beautiful predictions are ruined. In TMTB, because there are environmental fluctuations, there is some level of storage effect, and as a result, the predictions of TMTB are more robust. But if you want to ask me about it later, please do. So to summarize and conclude this part, I've shown that, and others have shown this before me, that random events of birth, death, and immigration can explain the static pictures of ecological communities, but not the dynamics. And when we introduce environmental fluctuations, this allows us to explain both the statics and the dynamics. Now, the idea that environmental fluctuations are of paramount importance to diversity and to dynamics are not new and often are attributed to Hutchinson. But I would still say that this is probably the first general and simple model that aims at and is capable of explaining both the richness, the commonness and rarity of species, as well as their dynamics, which means that perhaps it can serve as a first or second order approximation to ecological communities, real ones, that of course are more complex. I'll just finish this part with adding that after and pretty much in parallel to my work, others have reached similar conclusions and they have extended this work to other systems, also to other slightly different models. I'd be glad to talk about that also if you're interested. Okay, now that we've discussed neutral theory, I think it'll be interesting to go to even simpler models. In fact, the simplest model that you can have, I think, is the simplest dynamic model of an ecological community, known as dynamic equilibrium, or simply island biogeography. Importantly, why don't I call it by its name, island biogeography? Because I don't want you to think about islands. I want to think about any community, basically, that undergoes this very simple dynamics. What exactly do I mean by dynamic equilibrium theory? It's very convenient to formulate for a single species in the following way. If there's some species at time t, if it's present, it has some probability to go extinct and be absent by the next time step. And if it's absent now, it has some fixed probability to colonize and become present at the next time step. So present species go extinct, absent species colonize. Nothing can be simpler than that. Now, if you have many species playing according to this game, then we get the famous curves of species richness versus total colonization and extinction. And as most of us have learned in their intro to ecology, in the moment there are lots of species, then total colonization is low, total extinction is high, and species richness returns back to its equilibrium. And in the context of islands, this theory has made specific assumptions about how colonization and extinction depend on isolation and area. And with these assumptions, people have explained various static patterns, biogeographic patterns in islands and continents. But I don't want to go into this. This is pretty well studied and known topic, I want to try to understand whether this dynamics is in any sense sensible. And if not, then why? But before we move on, let's consolidate what's going on in the theory. First of all, what are the ecological mechanisms here? Well, basically, the theory asks, what happens? How will the world look like if species just would colonize at random and go extinct at random? That's it. Simplest you can have. It assumes, therefore, that the colonization and extinction rates are fixed and that the species are independent. These are the assumptions. And based on that, the theory predicts that richness will fluctuate around an equilibrium and the total magnitude of changes will be small. I'll quantify this more accurately later. And the composition of the community will show some turnover in time. Okay, so pretty much as soon as this theory was presented, people went to examine it, to test it with perhaps one of the most famous experiments in ecology, the defaunation experiment of Simberloff and Wilson, where they fumigated 6 trees that were islands for arthropods in Florida Keys. No arthropods survived. And then they looked how the number of species increases back as time goes by, how the islands get recolonized, pretty much returning to the same initial value of richness. And they wrote in their classical paper, the curves of figures, blah, blah, are believed to be explained by the following equation. And the following equation is just the mathy version of the story I told you with dynamic equilibrium. So yes, the theory support the experiment, very good one, very strong one, supports dynamic equilibrium. Another classical work is the work of Chair Diamond in the islands off the coast of California. There were 2 surveys of birds and of their richness and composition. And there were many extinctions, many colonizations. The composition changed quite a bit. At the end of the day, extinction roughly equals colonization. So richness was more or less fixed, and this supports dynamic equilibrium. Now, after the 60s, this topic of temporal changes in richness and composition has not been as hot until recent years, where there is a boom, this is BNAS in science, there's a boom of interest in temporal changes in richness and compositions, largely motivated by concerns over anthropogenic effects, of course. And okay, so this is what has been done. I do think that at least some of the works have a few important limitations. First of all, at least initially, the theory wasn't tested with many communities. It was narrowly applied, and also people haven't that much looked explicitly on the assumptions, because any violation, any deviation from the predictions has to stem logically from a violation of the assumptions. So not many people tested the assumptions, although they're pretty trivial. More importantly, I would say that some of the works have not been quantitative. Basically, you say you see a change of 4 species. Is it a lot? Does it reject the theory? Oh, perhaps it's too stable. You could have a too stable community. How do we know? More generally, if we have information on the composition at a presence-absence, 1 or 0 level, of a few species through time, and the corresponding figure of the number of species through time, how do we know if this is consistent with dynamic equilibrium, this pattern? Or I think even more interesting is not if it's consistent, what is the p-value? It's even more interesting to ask, what are the deviations? In what aspect does the dynamic deviate from dynamic equilibrium? And perhaps in some aspects it doesn't. And if there are deviations, then what are they caused by? These things have not really been studied. So the aim of this part of my work was to develop a methodology for testing the theory and measuring the deviations from the assumptions and the predictions. And we required that the theory would be quantitative, rigorous, robust to various problems in the data, as well as easily applicable. And having an easily applicable method, we wanted to perform an extensive test of the theory with as many communities around the world as we can put our hands on, their data, I mean. And finally, we wanted to explain the deviations. Basically, what processes are missing in this picture of random colonization and extinction of species, so that we would be able to explain the dynamics that we actually see in nature. So I will present to you the methodology. Basically, the moment we have this complex data, we need to summarize it in some meaningful way. So I will present you the summary statistics that we have developed. And then I will talk about the null model. The idea is to compare the statistics to a null model that resembles dynamic equilibrium to a large degree. I will begin by presenting the null model. The idea is that we need a way of generating synthetic species level presence-absence time series that would resemble the original data, but would be able to force the assumptions of dynamic equilibrium on the original data. So suppose this is the time series we saw the species was present in this year, present here, then here it got extinct, there was an extinction, so 3 years of absence, here it arrived again, we see it. And the first thing we do with this data is record its order, presence, absence, presence, absence. And then we preserve the order and the number of the colonization and extinction events, and the number of years of presence and the number of years of absence, but we reshuffle the timing of the colonization and extinction events. So possible resamples could be these ones. And you can see it's always presence, absence, presence, absence. In all this, in these resamples, always 5 years of presence, 4 years of absence, but the timing of the colonization and extinction could be anywhere. Now, because this null model is applied to different time series independently, it inherently generates independent time series. And also, very importantly, it can be shown that this algorithm generates time series that are approximately with fixed rates. And that means that the data that this model generates satisfies the assumptions of dynamic equilibrium and can be used to force these assumptions on the original data. Okay, so now we have this data from the composition through time of some community, and we have some synthetic communities that were generated by the null. What do we do with that? How do we look at this this big table of ones and zeros? Well, since the goal is to test the assumptions and the predictions, we've developed a set of statistics to test the assumptions and the predictions. You don't have to remember them. I will always write the interpretation nearby. If you lose me a little bit in the details, that's fine. Try to see the big picture. To test the assumptions of uniformity of extinction and colonization rates, we have developed the statistics of var_x and var_col. To test the assumption of independent species, we have computed the average covariance over pairs of species. And to test the prediction of small changes in richness, we have developed the DS-REL statistic. Most importantly, all these statistics are compared to their expectation under the null, and if they are constructed in a way that if the data was generated by dynamic equilibrium, then their value would be 0 on average. I would summarize many months of work and say that we have shown that this method is rigorous in the sense that it has power, it doesn't have a lot of, it doesn't have excess type 1 errors, it's reasonably robust to missing years, to cases of false detection of species, to cases where species are missed in the data. A lot of work. The bottom line, I'll try to give you a feeling of how they work. Suppose we have this species richness time series, and I want to know, how do the observed changes in richness compare to what dynamic equilibrium would predict? So observed changes in richness. Well, one way to quantify that is to find the maximum value of richness, the minimum value of richness, and subtract to get the observed changes. But now in the resampled time series, there are also changes between the maximum and the minimum richness. So I can subtract the expected change in all the resamples from the observed change. And it will be convenient to normalize this by average richness so that the statistic would be very comparable among different communities with different average richnesses. So at the end of the day, the meaning of this statistic is the excess richness changes, the changes beyond those expected under the null. If under dynamic equilibrium, we typically have a difference of 2 species and you get 4 observed, then this thing would be 2. Finally, of course, under the null, this thing should be 0. There would be no excess richness changes. Now, to test independence of species, Suppose we have these two species. You see I and J. When I is present, J is typically absent, and vice versa. Perhaps they're competing. Perhaps, when species J arrives, and species I says, this town just too small for the both of us. One of us has to go. And you see that, well, one of them goes. And a convenient way to quantify this is their covariance. In this case, the species had a negative covariance of these two time series. And now we can take every pair of species and compute their covariances and compute the average covariance over all pairs. And under dynamic equilibrium, since the species are independent, their average covariance should be 0. Okay, so this, you don't have to remember the details, just the big picture. And now that we have this machinery, this statistical machinery, do ecological communities around the world work like that? And if not, then in what sense don't they? I think a good place to start would be with the classical defaunation experiment of Simberloff and Wilson. And here again, we see the figures of the 6 islands that were in the experiment, the number of species through time. The blue is the observed, the red is the null, and you can see that at least in these 2 islands, the changes in richness are considerably bigger than what we would expect. In this case, this value of 0.67 means that, the observed changes are larger by the average richness times 6.68, which is substantial, 68% of the mean. Okay, now extending this to all of our 4 statistics, quantifying the assumptions and the predictions. Don't look into the numbers too much. In the 6 islands, you see, well, quite a bit of deviations. So I actually tried to go into the biology of this with Dan Simberloff's help. The data, it's always easy to compare to blame the data. The point is that, first of all, the suggestion that the curves of figures 1 to 3 are believed to best be explained by the following equation. And if this method works even in the classical experiment, the relatively short-term experiment that is supposed to confirm the theory, that means that the methods have power on a real data set. So let's try them on something bigger, the world. Not really. The idea is to, we decided to construct a large database of community time series from as many time series we could put our hands on, perform this test of dynamic equilibrium on them, and then try to explain the deviations that we find. Basically violating which assumption leads to the deviation in the predictions. So where do you find a lot of time series of ecological communities? A good place to start would be the North American Breeding Bird Survey, which is the single largest survey of biodiversity that's done in a single protocol. Another very good source is the BioTime, which is a newly available global database of biodiversity time series. There is information on mammals, grasses, arthropods, a lot of fish, a lot of birds, everyone likes birds. And yeah, from all around the world. And finally, an undergrad, Shiran, helped me gather some more time series into the big database. And how does this database look like? First of all, the basic thing that I can apply my method to is this, a time series of composition through time in a single community. So I call this a community. However, there are many cases in which there are similar communities. For example, all the North American breeding bird survey data, all these communities are, breeding birds in North America. So it makes sense to lump together these relatively similar communities into one data set. So the breeding bird survey is one data set, and there are multiple data sets, each having at least one community, and some of them have only one. And at the end of the day, I ended up with 5,000 communities in 49 data sets. 4 of these data sets were huge. They had above 200 communities, which is very nice. The breeding bird survey had 3,500 communities. And now I performed the analysis on 2 levels, and that's important. At the level of the data set, I've computed the average statistic on all the communities. And for the 4 data sets with 200 and above time series, I basically did the next analysis also for individual communities separately. So, do communities around the world follow dynamic equilibrium? And if not, then in what sense don't they? So let's look first at a histogram of the statistic of excess richness changes. As we said, under dynamic equilibrium, it should be 0. And it's not. This histogram describes the distribution of the statistic at the level of the data sets, the 49 data sets. And you see that there are pretty substantial excess richness changes in many communities. The changes are almost never too small. They're almost always too large, significantly so in 71% of the communities, and also substantially, like 0.5 means the change is bigger by 50% of the mean richness. That's a lot. What about the assumptions of the theory? Well, the species are not independent, and the rates, I would say, at the dataset level are not uniform. A lot of deviations, and perhaps most remarkably, there's almost never negative covariance between species. The covariance at the dataset level is almost always positive, and significantly so in 80% of the communities and of the datasets. Now, with the 4 largest datasets, I could look at the level of per community. So this is the histogram of the statistics for the breeding bird survey, the fish of the Skagerrak Strait, the Konza Prairie in Kansas, and the North Pacific Ground Fish Observer. And as you can see, the distributions of the statistics all over the place look pretty similar. There are lots and lots of deviations from dynamic equilibrium. Okay, so what? I think a very interesting thing to ask is, okay, we found that there are 2 large changes in richness. Violating which of the assumptions leads to this? And the natural thing to do would be basically do some plot of, say, average covariance versus excess richness. You see that they are pretty strongly correlated with each other. And wherever there's a large covariance between species, these are standardized values, by the way, wherever there is large covariance, also the changes in richness are large. So the covariance is the primary thing that determines excess richness changes, the main deviation from the predictions of dynamic equilibrium. Also, doing the same thing with a more extensive regression analysis, basically I want to explain the variability in excess richness changes. Why do some communities have a large, high excess richness change? Some other communities have a lower, high excess richness change. So the explained variable in the regression is excess richness change. The explanatory variables are the uniformity of colonization, extinction, and average covariance. And it's pretty obvious that the average these are standardized regression coefficients, and they are in both at the data set level and at the community level within the large data sets. Always the average covariance is very strong and significant, while the effects of the non-uniformity of rates is considerably smaller and often not significant. So again, covariance determines excess richness changes. Okay, we've crunched a lot of numbers. We've seen a lot of statistics. What does that actually mean to ecology? Well, as we said, we see a lot of large changes in richness and also in composition, but I haven't talked about this part. We see that the rates are not uniform, and we see that species primarily positively covary. What does it mean? Let's begin with the covariance. This positive covariance of species means that species are present together and they're absent together. They appear together and they disappear together. Now, if competition was very, very important between these species, then we would have negative covariances between the species. They would replace each other rather than arriving and disappearing together. The fact that we have a primarily and ubiquitously positive covariance, it means that for the changes that we see on timescales of decades in ecological communities around the world, competition is at the end of the day less important than the similar response of species to the environment. Different species respond in a somewhat similar way to the environment, and as a result, you see large changes in richness because a lot of species appear, a lot of species disappear, and so you have large changes in richness. And this is a primary thing we see. Importantly, we could have seen something else. If, we had a strong niche structure, there would be like 10 niches, and every niche would be occupied by just one species all the time, and species would arrive and colonize and go extinct and replace each other, but there would be a fixed number of 10 niches and 10 species. Then we would get a DSRL that would be very small, very negative. The excess richness changes would be negative, and the covariance between the species would be negative. But that's not what we got. What we got is that this scenario doesn't exist. Whatever, the main thing happens is species appearing together, disappearing together, showing large changes in richness. It's worthwhile saying that these conclusions were obtained with a new, rigorous, and quantitative, and easily applicable methodology. Now, in some sense, testing the dynamic equilibrium, a theory that is so simplistic, is a little bit funny. I think it's more interesting to look at it as viewing it as a baseline. The baseline is random colonizations and extinctions. In what senses, this is the baseline, in what senses do we have a deviation from this? So this is kind of the deviation from some baseline. And I think this, in this sense, this is quite a useful thing. Now, this is strongly related to the classical topic in ecology known as the community assembly or assembly rules. In 1977, Jerry Diamond published his work on assembly rules, where he showed that pairs of species of birds and islands off the coast of New Guinea, I hope I'm not wrong, never co-occur. And he, like, if this one bird is here, the other will not be. And he interpreted this as evidence for competition. Now, afterwards, and in many works, show that there's a prevalence of negative associations between species in space. So what these works have done is they looked at multiple communities in space and asked, do they resemble a random pattern? Well, what we did was to look at a single community every time and look at its assembly process and ask, does it resemble a random process? And interestingly, the results are conflicting. You have a negative association in space and a positive association in time. And I think it's worthwhile thinking about a synthesis. If you want to talk about this later, about my guesses about this, we can do it. So I would want to conclude this talk with going back to the research questions. I started by asking for the two most minimalistic models of community dynamics, neutral theory and dynamic equilibrium. First of all, can the model explain observed patterns of community dynamics? And the short answer to that is no. The second question is, what processes do we need to add to explain community dynamics? And here I think there is some consistency between the two parts of the work. Basically, if we have some ecological community, well, these individuals undergo stochastic birth, death, and migration events, random extinction, random colonizations. What do we add? What do we need to add on top of that to explain observed dynamics? One option is the details of competition between species. The other option is just their responses to the environment. And I think both parts of my work indicate that if you want to explain the dynamics, the changes that you see on timescales of decades, what you would need to incorporate is the responses to the environment first and foremost. And I think it's also worth thinking that, again, looking from a temporal perspective, looking at these dynamic temporal patterns allowed us, I think, to shed light on processes that otherwise would be difficult to see. And with that, I will finish. I want to thank a lot my supervisors, both Ronen Kadmon from the Department of EEB in the Hebrew University and Nadav Shneer from the Department of Physics in Bar Ilan University for all the numerous things I learned from them, the Kadmon Lab and the Department of EEB at the Hebrew University, as well as collaborators, my funding sources, and wonderful data sets. I like data. And with this, I'll be glad to thank you very much. And of course, thank you very much for listening to all of that. And I'll be glad to answer questions. Thank you.
Thank you, Michael. And if people have questions, you can either put that in the chat box or you can unmute yourself and say it verbally.
I have one question. Can you hear me okay? Sorry, I have
Yes.
these headphones on. I wanted to ask about in the first part, when you add environmental fluctuations, are you effectively adding niche differentiation through storage effects? Is that a way we should think about why the dynamics change?
Well, the short answer is yes. This is in a sense, this is a merging of one of the ways to merge neutral and niche models. Basically, definitely, the moment all individuals of a species respond in a synchronous way to something, it means that there is some environmental factor that shapes them. Absolutely. So in some sense, this is incorporating niches. I don't think, honestly, I think that the differentiation between niche and neutral is kind of so it boils down to at some point to definitions. At the end of the day, in this case, yes, you can view it also as a niche model that has storage effects. Yes.
Okay, there is one in the chat box. So from Arno. So in the first part of the presentation, I'm not sure to have understood the way the parameters tau and the fitness distribution parameter are estimated.
Okay, thank you very much for the question, Arno. Well, I haven't described that. I didn't want to go into the technical details. The idea was the following. I tried to make the smallest number of arbitrary assumptions and things that could change things. First of all, there is another important parameter in this model that I haven't mentioned. It is basically the translation of time from years to time steps. Time steps in neutral theory works with time steps. The real world works in years. So how many years are there in a time step? This is easily to see, is you basically see how many individuals die in 5 years, which is the time between surveys. So now you know how many events took place. Next question, how do you estimate tau? To estimate tau, I basically asked, okay, I have, forget about the abundances. I have trajectories of species. They go up, they go down. How long on average does it take them to change their trajectory from up to down? The answer is, by the way, 10 years in BCI. And finally, the A is the parameter that needed to more fitting. There, I basically tried to fit the magnitude of fluctuations and another pattern that I haven't shown you, the scaling of fluctuations. I think it's even a more interesting pattern, but it's more complex technically. And then I just picked the A that works best. And the other parameters are just exactly the same as in neutral theory. M is 0.1 and theta is 50. Just as people have done that before me, nothing changed here. So that is to you, Arno. I'd be glad to, if you're interested in this kind of things, I'd be more than glad to talk about it afterwards.
Okay, another question from George Kling. Thank you, Michael. In the plots you showed with covariance and DS, the number of positive and negative DS values were roughly equal. But in a histogram you showed, you made a point that there is mostly positive DS, not negative. Why there is a difference? Is it because you standardize value, say Z value, for the plots of covariance and DS?
Thank you, George. You're absolutely right. These are standardized values. It means that I subtract. there are lots, the plots would not look the same if I wouldn't standardize. But the moment you standardize, the moment you subtract from every variable its average and divide by the standard deviation, they look the same. It also makes it more comparable between different communities and different systems. This thing is based on standardization, so you have roughly the same numbers. Yes, so you got it right.
You were also alluding at the end of the talk in this conflict between the spatial negative covariance and the temporal positive. So what are your thoughts about that?
Well, I can throw a guess. At some point, it could be that there is filtering, basically, that the species that make it, that the species that you actually see in time, and so you can only do this analysis in my approach with the species that you actually see. If you haven't seen them in a specific location, they are not in the game. And as a result, I think that probably there are some filtering factors, biotic or abiotic, that just prevent completely some of the species from arriving to some places. And then you see negative associations between species. And then the species that arrive could be already similar enough so that they would respond similarly to many, many environmental changes, not necessarily to all environmental changes. Also, the fact that environmental changes is so important to dynamics on timescales of decades doesn't mean they're very important for coexistence on timescales of tens of thousands of years. It's possible that for the competition structure is much more important to determine how many species will there be in 10,000 years, and in decades, everything we see is noise. Possible.
I'll ask you one more question about the niches that you added. Is there a way of thinking about how much of that you added? In other words, I guess how much stabilization or
In neutral theory?
yeah.
First of all, the moment you're assuming they're okay. In this very simple theory model, I assume they're independent, that the fluctuations of different species are independent. So probably you have as many niches as species. By the way, I think Chesson himself, that proposed storage effect, had a paper saying that you don't need niches anymore because we have the storage effect. So there's some ambiguity there also with him. How much, first of all, you can compute the invasion growth rate in this model. Is it very interesting? That's a different question. Basically, with this approach, you can just see how many species you have. And also, by the way, you can see the invasion growth rates in an indirect way in these figures. Rare species, it takes whenever you introduce some regimes of environmental fluctuations, rare species take longer to go extinct compared to the neutral case. That is basically the growth rate when rare, the storage effect introduces. Notice that it happens in the regimes where tau is, okay, tau is autocorrelation. Here, the autocorrelation is relatively short. The autocorrelation is crucial, but that would not reflect on your small r. And there is the new paper in Ecology letters explaining exactly the problems with invasion rate when rare. And actually, in some sense, you can already see that here, that invasion rate when rare is not affected by tau, but the time it takes an invader to go extinct does depend on tau.
What about if you turned immigration off in the model? Are there some species that would persist for a very long time? Or
Yes. Yeah, it's a model with stabilization. So yes, if you add stabilization to a model,
Yeah,
some species will persist very, very long.
I guess that could be one measure of how many niches you've added or something. Yeah. I think I see that your PhD advisor joined us for some of the talk.
It was, I think, most of the time here.
Yeah, good. Yeah.
Okay, so it seems there are no more questions. So let's thank Michael again for giving this very excellent talk today. And also thanks everyone for joining us today. And I'll see you next time.
Thank you very much.
Mhm.